So we've arrived now at the last major organ of the digestive system, and that is the large intestine. The large intestine extends all the way from the ileocecal junction. That's where the small intestine joins the large intestine to the anus. And the large intestine has a couple different pieces. It has the cecum, that's the first section, the colon, which is broken down into three smaller sections, the rectum, and the anal canal. The large intestine is all about just reabsorbing water and various salts, as well as dealing with any sort of microorganisms that may be in this section of the GI tract as well. So the large intestine starts out with the cecum. That's this little pouch right here. And we have the ileocecal valve that deposits the chyme, which will become feces, into the first section of the large intestine. You then have four pieces that make up the colon section of the large intestine. You have the ascending section, the ascending colon, which is the part that ascends, a transverse section that goes across, the descending colon, which comes down, and then this sigmoid colon, which is this S-shaped right here that I'm running my mouse over. So four pieces of the colon. And all along the colon, you have these muscular bands that are called teniae coli. When these bands contract all along the large intestine, they create these little pouches that I'm running my mouse over. These pouches are called hofstra. It sort of gives the large intestine this sort of puffy look to it. Then all along the large intestine as well, you have what's called omental appendages. These are these little fat deposits in yellow that I'm running my mouse over. So those are all lined throughout the large intestine. And then within the actual mucosa and submucosa layers, you have crypts. So these have glands with a lot of your familiar cells you've seen from the small intestine, such as your goblet cells, your absorptive cells, and your granular cells. So then towards the end of the large intestine, you have the rectum, which is a very strong muscular tube that leads to the anal canal, where feces at this point will be excreted from the body. And you have two sphincters in the anal canal, an internal one made of smooth muscle and an external one made of skeletal muscle that are under voluntary control. So now looking a little deeper in the histology, with those crypts, you have the crypts down here in the mucosa layer, which have your goblet cells that secrete mucus that help protect against any sort of acids or any compounds that came from the small intestine, as well as keeping things lubricated to keep moving along through the tract. Then you have your absorptive cells, which help, of course, absorb water and salts and other ions. And then your granular cells, which help fight against bacteria and other infections. Of course, you do have a lot of bacteria within the large intestine that are there for helpful reasons, helping to break things down and keep the large intestine actually clean. So bacteria themselves in the large intestine have metabolic byproducts. These are basically just acidic components. But there are also secreted by bacteria what are called antiporters. And these are going to exchange ions across the membrane of the mucosa to ultimately help remove the acid made by the bacteria. Additionally, bacteria help with making vitamin K, which is going to be reabsorbed by the large intestine. Vitamin K has a very special job in that it helps make a lot of your blood clotting proteins that help, of course, form the blood clot when you have a wound. And then bacteria make what is called flatus, which is just flatulence or gas. So then after all of these secretions and reabsorptions have occurred, what's left is feces. This is just any water that's not reabsorbed, any undigested materials, as well as any other microorganisms that are being excreted from the body. So within the large intestine, you have a couple different movements. The first type of movement you have are just called mass movements. These usually happen after meals. So if you eat breakfast, it's pretty common to have a bowel movement after that because of that stimulation you have from the meal. Then you have local reflexes. You have what are called gastrocolic reflexes and duodenocolic reflexes. Gastrocolic reflexes are just a movement in the large intestine when there's food in the stomach. So that signals the large intestine to then keep moving things along. because more food's going to be there soon. Same idea with the duodenocolic reflex. Having food in the duodenum is going to signal the large intestine to move things along. Then, of course, you have the process of defecation or pooping in order to excrete feces from the body. And this is signaled to an individual by what's called the defecation reflex. This is just when the rectal wall gets distended by feces. So it starts to stretch that tissue, which sends a signal to the brain saying, okay, it's time to use the bathroom. And of course, the process of defecation is voluntary based on those sphincters that we have voluntary control over in order to expel the feces by contracting the abdominal wall. So that's the end of the actual digestive structures. The last thing we're going to talk about is just the general processes of digestion of absorption for all of the macromolecules that we have. So your carbohydrates, your proteins, and your lipids, as well as a little bit of water and salts. So to recap a little bit, digestion is just the breakdown of food molecules for absorption. So with digestion, we have mechanical digestion and we have chemical digestion. Mechanical digestion, remember, is just breaking down large food particles into smaller ones. They're still the same chemical compositions, they're just smaller pieces. So when you chew your food, that food is still the same food, it's just now broken down into smaller pieces. It's not any different chemically. When we do chemical digestion, however, we're breaking the covalent bonds that make up the actual macromolecules. So for example, you have polysaccharides, which have glycotic linkages that link monosaccharide to monosaccharide to monosaccharide. When we break those bonds, that polysaccharide will become eventually a disaccharide and then a monosaccharide. We're trying to break down that bigger macromolecule into its little pieces to absorb. Then when we do that, we of course have it be absorbed through either the small intestine with the duodenum or the jejunum. Then we have it transported in the bloodstream. So first major group of macromolecules are your carbohydrates. To recap this from intro bio, you have your monosaccharides, which just are one sugar, things like your glucose, your fructose, and your galactose. Those are the three monosaccharides. You have your disaccharides, di being two, so just made up of two sugars. And then you have your polysaccharides, which is just meaning many, so many sugars. And we have digestion of carbohydrates in a couple of different places. First one is the oral cavity. The salivary glands produce what's called salivary amylase. This breaks down initially some of your polysaccharides in the oral cavity. Then we don't actually have any carbohydrate breakdown until we reach the small intestine, where we have some of those pancreatic enzymes get released into the small intestine to help continue to break down those carbs. So we have pancreatic amylase, which helps with carb digestion, and then we have the disaccharidases, which break down specifically disaccharides into monosaccharides. Then to absorb those carbohydrate compounds, they vary based on what monosaccharide we're looking at. So your glucose and your galactose are going to cross that intestinal membrane by what's called symport. Symport just means that another compound or ion has to move across the membrane with it to allow that molecule to move. So in this case, we have sodium move across, and that allows us to also move a molecule of glucose or a molecule of galactose as well. Then for your fructose, that goes by what's called facilitated diffusion. A facilitated diffusion is just moving a molecule through a channel protein. It can't readily diffuse from one side to the other, so it has to go through one of those special channel proteins to make it through. So show you a little diagram of how this works. We have your glucose molecule that is a form of monosaccharide, and it goes across through symport of sodium. So sodium comes across the membrane, and that allows glucose to move across as well. This is powered by a sodium-potassium pump, which allows this process of symport to happen. Then when the glucose is inside, it's going to go through facilitated diffusion by moving through one of your special channel proteins into the bloodstream, where that will then be carried off to the liver to be processed. Next, we have your lipids. So your lipids, there's a bunch of different classes of them. You have things like your triglycerides, your phospholipids that make up the membrane, steroids that make up different types of hormones, as well as fat-soluble vitamins. So lipids get digested in a couple different places. In the oral cavity, we have a special enzyme called your lingual lipase. In the stomach, we have your gastric lipase. In the pancreas, the pancreatic secretion is your pancreatic lipase. So they always end in lipase, letting you know that those break down lipids. And then also, we have the process of emulsification that occurs in the duodenum through bile salts, which are part of bile. Bile salts are able to turn those larger fatty droplets into smaller droplets that are called micelles. When we bring those big lipid droplets into smaller lipid droplets, that increases the surface area and allows them to be more easily absorbed by the small intestine. So when those micelles are ultimately absorbed across the small intestine, those lipids, whether they be triglycerides, phospholipids, steroids, whatever, are going to be repackaged into what's called a chylomicron. And these are basically just protein packaging of the fatty tissue or of the fatty acids. So show you a diagram for that. We have an example of a fatty acid or a monoglyceride, whatever we're looking at. And that gets emulsified by the bile salt. So it breaks it down to a little droplet. That droplet then diffuses across the membrane and gets inside wherever the intestinal cell is. We then have that new protein coat go on top of it called the chylomicron. And then through exocytosis, it's going to exit the intestinal cell and not go to the bloodstream, but actually go to that special lymphatic vessel called the lacteal. Eventually, however, the lymphatic system will deposit those lipids into the bloodstream. In order to be carried through the bloodstream, they're going to need to dissolve in the blood plasma. To do this, they're going to have to form a compound that's called a lipoprotein. Lipo just being the lipid component, and protein just being the protein component. So lipoproteins are needed to carry them through the blood plasma. One example of one of the fats that can be transported in this way is your cholesterol. And based on what the compound is, it can be transported in three different manners. You have your very low density lipoprotein, your low density lipoprotein, and your high density lipoprotein. So your very low density lipoprotein and your low density lipoprotein are known as bad cholesterol because they have a much higher lipid concentration relative to the protein concentration. The density that comes from these names for low density and high density has to do with the amount of protein content. So for your very low density, your protein only has 8%, and for your low density, it only has 25%. Since proteins are heavier than lipids, that means they have more mass, which ultimately means they have more density. So the density is based on the protein content. So for very low and low, we have much lower protein contents for both. So like I said, these are the bad cholesterols, and ultimately these bad cholesterols can build up in the arteries and ultimately cause atherosclerosis, which is the buildup of plaque, which can cause things like heart disease and stroke. Then you have your high density lipoproteins, which have a much greater content of protein relative to the lipids. And ultimately, these forms of cholesterol are able to just go back to the liver and be flushed out of the body. They're considered the good cholesterol, quote unquote. Ultimately, however, the low density, high density, or very low density cholesterol is going to be based on dietary choices. So what you're actually eating, how fatty it is, and what kind of fats it has. Okay, lastly, for our macromolecules, we have your proteins. So your proteins are built based on what are called peptide chains. So based on the proteins is how many peptide chains they may have, and those peptide chains are then broken down into amino acids. So digestion for the proteins occurs in the stomach and the pancreas. In your stomach, you have that compound pepsin that gets converted from pepsinogen, remember, by hydrochloric acid. Those break down your proteins into the smaller polypeptide chains. And then in your pancreas, you have the pancreatic proteases, And ultimately, these make dipeptides, tripeptides, and amino acids. So a dipeptide just means there's 2 amino acids. A tripeptide just means there's 3 amino acids. And then an amino acid is just an individual amino acid. So the idea is we're getting them down from the proteins to polypeptide chains to very short polypeptide chains or individual amino acids to increase the ability to be absorbed. And for the pancreas, with those proteases, you have your trypsin, your chymotrypsin, and your carboxyl peptidase. So for absorption of proteins, ultimately how it's going to be absorbed is going to be based on the pH. So the acidity level of the actual amino acid or peptide chain length. And the pH is going to be based on then the charge of the amino acid. You don't have to specifically worry about that, but just know that pH is going to dictate the transport. So for your compounds that are acidic and neutral, these are going to be co-transported with sodium. And then for your compounds that are more basic, so a pH higher than 7, these are going to get into the intestinal cells by facilitated diffusion, going through those channel proteins. And then ultimately, after they're absorbed, the amino acids are going to go to the bloodstream and into the liver to be processed. So showing the same diagram again the third time now with proteins, we have your amino acids, which in this case is an acidic or a neutral compound because it's going to go across the cellular membrane by way of symport with your sodium, again powered by that sodium-potassium pump. Once it's actually in the cell, it's going to go through facilitated diffusion and enter the bloodstream to go be processed through the liver. If this compound were basic, however, it would then just enter the cell through a facilitated diffusion through a special channel protein and go through the same process, entering the bloodstream and going to the liver. So to recap all of that, since that was a lot with the different types of digestion and absorption, with all 3 compounds, we have your carbs, your lipids, and your proteins. So your carbohydrates in the mouth are broken down by salivary amylase into polysaccharides or disaccharides. Nothing happens in the stomach with carbohydrates until we then get to the duodenum, that first section of the small intestine, because we have secretions from the pancreas and the liver here. So for carbohydrates, we have your pancreatic amylase, and that breaks those compounds down into disaccharide form. And then we have those disaccaridases, which break them down into monosaccharides. So then for your lipids, in the oral cavity, we have your lingual lipase, which does some initial breakdown of lipids. In the stomach, we have your gastric lipase. And then in the duodenum of the small intestine, we have your bile salts in the liver, which help emulsify fats. Your pancreatic lipase is created, of course, from the pancreas. And then your cholesterol esterase, which we didn't really talk a lot about, but that has to deal with cholesterol. But the big ones are bile salts and pancreatic lipase. So ultimately, we're trying to break this down into fatty acids, monoglycerides, and then cholesterol. Then for your proteins, nothing happens in the oral cavity until we reach the stomach, where we have initial digestion by pepsin. So that will break down some of your proteins into polypeptides. Then we get to the duodenum, and several of your pancreatic enzyme secretions, trypsin, chymotrypsin, and carboxyl peptidase, break down those polypeptides into shorter peptide lengths. Then further along in the small intestine, your peptidases break down those peptides into either individual amino acids, dipeptides, which are just 2 amino acids together, or tripeptides, which are just 3 amino acids together, to be again absorbed across the small intestine. So lastly, we have your water and ions. So water ultimately is able to move in or out of the small intestine via osmotic gradients, which just means that we're looking at high concentration of water to low concentration. Same idea as the pressure gradient we talked about with the respiratory system going from high pressure to low pressure. In this case, we're just looking at water concentration, but it always goes down that gradient from high to low. And ultimately, you have a lot of different sources of water throughout the body that inject water into the GI tract. Naturally, we ingest water by food and drink. We also have secretions from the salivary gland, the stomach secretions, the liver bile, and the pancreatic secretions, as well as any secretions from the small intestine. All those go into the digestive system. However, to make sure we're not excreting too much water beyond what is needed, most of that water is actually going to be reabsorbed in the small intestine, so around 92%. And the rest of that water, while that feces is being formed in the large intestine, is going to be absorbed across the large intestine epithelium. So then when you actually have the excretion of the feces, really only 1% of the water content is actually being excreted. The body is very, very efficient at reabsorbing water. Then with your ions, so things like your sodium, potassium, calcium, etc., these are going to be actively transported across the small intestine wall. Active transport means that we need to input energy into the system to get them across the membrane. If we don't have energy, we don't have any sort of transport. And of course, we need to absorb these ions because things like your sodium, potassium are needed for action potentials all throughout the body. Calcium is needed for blood calcium levels with your bones, as well as muscle contractions. So your enzymes are very, very important. So they have to be readily uptaked by the body. And then your chloride ions, also involved a little bit with your action potentials, are going to move passively across the small intestine membrane. So they can just diffuse from the inside of the small intestine through the membrane. Okay, so that will pretty much wrap up this chapter on the digestive system. I will see you next time for the urinary system.